This experiment demonstrates in ovo electroporation of chick midbrain to study the effect of exogenous gene expression on midbrain development and dopaminergic neuron differentiation. First, inject the midbrain of Hamburger-Hamilton stage 11 chick embryos with desired plasmid DNA to specifically target exogenous DNA into the developing midbrain vesicle. Then, using custom-made small electrodes, electroporate the midbrain vesicle to deliver the exogenous DNA specifically into the midbrain neural progenitors. Next, harvest stage 23 embryos with good M-cherry expression in the midbrain and generate optimal midbrain transverse sections. Ultimately, immunostaining analysis for dopaminergic neuron markers, such as tyrosine hydroxylase, can show the differentiation of neural progenitors into mature dopaminergic neurons induced by the expression of genes of interest.
The main advantage of this technique over other existing methods of in ovo electroporation is that this protocol can achieve midbrain-specific electroporation to study midbrain dopaminergic neuron differentiation.
Though this method can provide insight into midbrain development, it can also be applied to other parts of the nervous system for performing fate mapping analysis and for investigating the regulation of gene expression.
In order to acquire Hamburger-Hamilton stage 11 embryos, position each fertilized egg on its side and mark the top with a pencil dash. Then place the trays on a rocker in a pre-warmed, humidified incubator. Check the humidity and water level every 24 hours. At 41 hours post-fertilization, remove the eggs from the incubator and store at room temperature.